Gallaudet University presents.
Good afternoon, everyone. It's very nice to see all of you here. This is our first lecture of the spring semester. This afternoon, we are very privileged to have with us Dr. Kenneth Pugh. This gentleman right here. Dr. Pugh has a very long and exciting record of work in the area of neuroscience and cognitive sciences. He has served in many roles, and I will list some of what those roles are. Most of us have problems with the one jobs that we have, but he's got 4 or 5. He's the president and the director of research at the Haskins Lab. He also has an appointment in the Department of Psychology, where he is a professor at the University of Connecticut. He's also an associate professor in the Department of Linguistics at Yale University. He's an associate professor in the Department of Diagnostic Radiology at Yale. He is the director of the Yale Reading Center, and he is the co-leader of the Yale Haskins Teagle Foundation Collegium on student learning. He's also a principal investigator of a number of grants, and he is very well published in the area of cognition and cognitive neuroscience. He is a true pioneer in work that has led to our understanding of brain functioning and brain structure, as well as linguistic development. And he has published in a number of very well-recognized, prestigious journals. He has won a number of awards. In short, I can't say enough about him, and you can understand why we are so lucky to have him with us today. He also was recently appointed to the advisory board of VL2, and we are delighted to have him working with us in that capacity. I could go on and on and on, but that would take up more of his time. And I think it would be better for you all to see what he has to say rather than to see me continue on. So with that, I will turn the floor over to Dr. Pugh. Ken?
Thank you. Thank you very much. First of all, I want to thank Dr. Allen, Dr. Petito for inviting me. It's really an honor and a privilege to be here today and meet with you. I was very lucky this morning to have a tour of the campus with a very bright young student who knew everything in the history and was giving me a wonderful tour. And I'm just amazed to see all of the history and to know that it goes back to Abraham Lincoln. I didn't know any of these things until I came today. I'm really thrilled to be here. I also want to make a note. Dr. Allen mentioned that, my work had been somewhat pioneering in reading disability, but I want to acknowledge a dear friend who we don't see enough of each other anymore, Dr. Guinevere Eden, whose work and my work in the old days when we were kids, starting out and looking at brain and reading and reading disorders, influenced mine, and I think we influenced each other in a lot of good ways. So it's really a pleasure to see you, Guinevere, again. And I want to thank all of you for coming. And I want to especially thank the translators. Now, I'm from New York, and Laura Ann knows what that means. And I tend to talk fast, so I'm going to slow it down. I'm going to be careful in making sure that everything gets appropriately translated and clear. And I will also make sure that I complete my talk in leaving enough time for questions and discussion, because I will be covering a lot of material, and some of it is very complex and technical on the surface. But this isn't really a talk about the details of imaging technology. And my job here is to really try to convey why these technologies and approaches to understanding typical and atypical language and reading development are important and useful. And in sort of, there's always two parts of it that have to be discussed when talking about the neuroimaging and all the excitement that surrounds it. One is the promise and the utility of this, which is great. And the other one is avoiding the hype, which is also great and sometimes inappropriate. There's much that we've learned with studying the brain basis of language and cognition, and there's much we haven't. And there are some limits on what we can get in any one tool, with any one set of tools. And at present, even the best of our work on reading disorders and their remediation is still in many ways descriptive of the system rather than explaining why it does what it does. And I'll try to talk a little bit about that as we go through the talk. So it's really important for cognitive neuroscience, which is this interdisciplinary field that links psychology and other fields to neural pathways. It's very important for us to go beyond description and toward explanatory, testable theories. And I want to talk to you a little bit about new data from a longitudinal study that we're running that at least begins to try to get these data. I also want to bring greetings from my colleagues at Haskins Laboratories. As Tom said, we're at Yale University, but we're an interdisciplinary independent institute. We've been around for 75 years and always exploring at heart. The sort of core mission is to understand the biology of human communication in all its forms. This includes speech and reading and signing. And over the years, we've had a number of researchers who've interacted with Gallaudet, and one who's now a professor here, Gaurav, who was a postdoc at the time I met him. We have always been interested in understanding language in all its manifestations and forms, and in different in different orthographies and different representations. And I'm very interested, I must tell you, and then I'll end this introduction, which is going on a bit. I want to tell you that I am particularly interested in learning more about sign language and about deafness and language and the link to reading. My area is reading. And I'm just kind of thrilled to be here and able to be on the advisory board and think about research ideas going forward on this. So anyway, thank you all for inviting me. The focus of my talk today, they're going to try to get a pointer, but I'll for now point to the screen. I'm going to present this work, which is entirely on hearing children. And unfortunately doesn't have any data, at least from my lab, on how some of these brain behavior relationships that I'm going to explore might differ and might be importantly different in kids who are learning sign as a primary language and then learning to read. But I hope that what I can share with you today is this. What are the lessons we've learned as a field on reading and its disorders in hearing populations? What are some of the brain systems that seem to be critical for the development of reading? What are some of the patterns we see when we try to remediate reading in hearing populations to provide, I guess, a background and a foundation against which to ask important questions on how that would differ in sign populations. And indeed, I would note that a lot of the work on the brain basis of reading and reading disability, early on at least, was done in English. And over the last 10 years, there's a growing appreciation. Guinevere's lab, our lab as well, are interested in trying to understand what's universal, even in written languages, in hearing population. What are universal factors that underlie risk for reading problems in any orthography, whether it be alphabetic, like English, or whether it be any other written form. And so in my research, and at Haskins for many years, we've been looking at the development of reading and the brain basis of reading in different orthographies. And more recently, trying to get a better understanding of what happens when you are learning two languages and trying to create a literate knowledge of both. And this is where I'm particularly interested in sign and English reading as a bilingual challenge. So I really have much to learn, and I will share what I know, and I'll be back to learn more as I serve on this advisory committee. In any case, I'm going to talk about these topics, an overview of findings on brain organization for typically and atypically developing learners, and I'll talk about what we have seen to date with respect to the brain patterns that discriminate non-impaired and reading disabled children and adults. The second area is an update on some longitudinal studies that we're conducting that are trying to look at the precursors or the early factors that seem to be associated with a good trajectory for learning to read or a not so good. We'll also take a brief look at the end at reading remediation studies and what we sort of can conclude from current studies on plasticity or remediability in reading disability. And then I'll end by talking about next steps in our research and others in terms of getting at gene, brain, behavior pathways. So again, with the caveat that I'm talking about data that comes entirely from hearing populations, let me begin the formal presentation. For the vast majority of children who present with reading difficulties in alphabetic languages, there are pronounced difficulties in phonological processing, operating on the spoken elements that make up a language. And these phonological processing difficulties include problems in becoming phonologically aware or understanding that spoken words are made of smaller units. Problems in phonological working memory, holding things and operating upon them in a phonological form. And problems in speed of phonological processing, as indexed on things like rapid automatized naming. So in this world of reading and reading disability in hearing populations, learning to read their native tongue, if it's alphabetic in particular, phonological processing is a nearly universal feature of what presents when children come in with these struggles. Not all children, but the vast majority. What are the underlying causes of phonological deficits? Well, many of us, Guinevere and I and others, have gone after this for a lot of years. There is one line of research that assumes and tries to get evidence for the idea that phonological deficits, processing on spoken language deficits, are uniquely associated with language, and therefore search at the level of phonological representations or language-specific systems to understand the deficit. That is, the deficit in phonology is language-based and not reducible to something else, something more basic in processing. And work has proceeded along those lines for many years. There's also a number of researchers, including Guinevere and John Stein and many others, who've tried to make the link between phonological problems operating on the spoken form, and more basic deficits either in visual processing or auditory processing or other things. So for example, work by Stein and Eden and others has looked at both visual processing and visual motion processing as a potential factor underlying the development of later difficulties in phonology and in reading. Folks like Paula Talal have focused on the rapid changing auditory features that are sort of characteristic of the range you see in speech as potentially a source of auditory deficit that might impact and presage difficulties in phonological processing on speech and subsequent problems in reading. There's also more recent work by Usha Goswami and others that also look to auditory processing, but not in the rapid high frequency range, but in lower frequency range that might be more associated with amplitude detection deficits. And so all of these areas are being mined and explored to this day as potential elements in basic sensory motor processing that might be contributing to the well-agreed phenotype of phonological processing deficits. There's also some interesting work by Ramu and others on problems in accessing and operating upon the different kinds of phonological information that need to come together to become phonologically aware. And more recently, there's actually a somewhat interesting line of studies coming out of people like folks like Anne Sperling and to a degree Joe Ziegler recently that have tried to identify problems that I wouldn't want to call them attention, but they have to do with signal noise difficulties in low-level processing. So in sort of difficulties in amping up the signal and reducing noise, or problems with what Sperling calls noise exclusion. So these are all out there as potential etiological factors. There's also a more basic question that, or a more basic view that we're starting to pursue that lies in the language domain, but in much more the mechanisms of listening and speaking. Again, my research focuses on hearing populations, and so the appropriate caveats for what goes on research here. But there is evidence that young children who will present with reading problems later on show subtle difficulties both in perception of speech and in producing complex utterances. And what we know about the pathways that are associated with perception production learning in speech suggests that some of those pathways in the brain are already implicated as potentially showing reading-related deficits or differences. And so one area that needs to be understood is how problems in perception and production themselves, and we can't underestimate the role of production, might be associated with difficulties in plasticity and reorganization of the brain in order to become able to connect specialized systems for language with visual representations. And so this is a lot of detail. It may go beyond some interest in the audience. It may be closer to others. But there is a lot of discussion about mechanism. And then there's the always annoying but often seen reality in biomedical research that, in fact, there may be many roads to Rome or multiple pathways to a common end state, and that some kids may, in fact, be differentially presenting these difficulties and ending up with phonological difficulties that make learning to decode difficult. Thank you very much. I appreciate it. Great. Thanks, man. So all of this is the landscape against which we begin to explore the brain in reading development and try to identify the causes of reading disability. And the argument made from neuroscience and from cognitive neuroscience is that each of these different models or speculations are associated with very different brain pathways, which may or may not be differentially compromised in kids with reading disability. And so the bottom line is that mapping into the brain in order to understand the mechanisms that underlie these observable phonological deficits is in fact both warranted and exciting at the same time. Now, there's also some consideration as we begin to think about the brain. 4 seconds, yes. Great. There's some consideration that we have to have about the difference between natural language communication and reading and writing in terms of how much brain specialization there might be. It's interesting, guys, because when I present this slide in other talks, I've often written spoken language is a biological specialization, but reading and writing are relatively recent cultural inventions. In other words, we're wired to communicate by perception and action. Note that I took spoken language out because when I came here and I realized that I need to think about this in a very different way, whether we're talking about language through manual gesture in its communicative natural form, perceiving and producing, or speech, we are talking about language. And as Laura's work has shown in babies developing signing, there is a naturalness. You don't need to go to school to pick up all the information. The brain is coming ready to perceive and to produce, to communicate. And there's a good deal of brain specialization for language that it appears signing taps into just as speech does in hearing children. But reading and writing, however it goes, is a relatively recent cultural invention. It's only been around for 5,000 years, and it is a very different form. It is a way of representing language, whatever language is, by visual symbols. And of course, it involves taking lots of brain regions that have evolved to do lots of jobs, vision, language, associative learning, memory, attention, and transforming them into a reading circuit. In simple statement, there cannot be brain specialization for reading itself, but rather reading is an exercise in neuronal recycling, as Dahan likes to call it, or in broad neuroplasticity, or in retraining neuronal systems to a new function. And this is the practical implication of all of this. While language is learned naturally in the vast majority of children, reading is, in fact, requires from almost all children direct instruction in the nature of the rules by which orthographies are built. And significant numbers of children in all languages will fail to master reading. It is a real cognitively challenging reorganization of the brain, which, by the way, implies that if you're looking at any population, you will see this phenotype of reading disability present. And how it would present in signers who are learning to read, I think, is a fascinating question. And you can see I'm putting in a plug for trying to do that kind of research, and I'm interested in being part of it. So we're arguing that we need to go to the brain, that there is value added. Oh, yes. Good. I'm arguing that we need to go to the brain, that there's value added in understanding the neural systems for language and reading and how reading develops in order to get information that might not be completely available to behavioral studies alone. So what do we know about language, reading, and the brain? Well, let me start with the classical approach, exemplified beautifully, by the way, in the work of Norman Geschwind, who really organized existing knowledge about lesions and injuries, accidents of nature, affecting the brain and changing both speech and reading. And from this work, we start out with a very simple model for speech and reading based on this lesion research. That is, studies of patients who have had injury to some piece of brain and have lost some function. And the very simple classic Wernicke's, Geschwind-Wernicke's Lichtam model is one that looks like this. We have specialization for language along the perisylvian plane, including Broca's area, which is broadly and somehow generally associated with motor activity and production in some generic sense, and Wernicke's area, which is broadly and generally involved in comprehension of language in some sense, and the connective pathways like the arcuate fasciculus that link these perception and production hubs. With regard to reading, Geschwind looked at the existing lesion literature, which identifies the angular gyrus in the inferior parietal cortex as an area which, if damaged, produces an acquired dyslexia, and speculated that the visual information on letters gets into the language system because whatever your language is, whether it's visual or whether it's oral, the job of a reading brain is to get as fast as possible away from vision and into the biological hardware for language, syntax, and comprehension. And in fact, a skilled adult reader in any language, if things go well, gets from vision to phonology and meaning in a couple hundred milliseconds and change. So the brain needs to solve the puzzle, how to get into language and away from the visual form as quick as possible. What are the brain pathways that allow you to be able to recognize words in a couple hundred milliseconds so that you can process these visual representations as sentences and not tax working memory and cognitive processing? How do you make reading like listening in the case of hearing populations? So his initial model is, I would say, not entire, it hasn't gone away even with all of our modern cutesy neuroimaging studies identifying different hubs and different pathways. Still, this basic framework stands up. But one of the problems with lesion studies, and there are a number, is that, for one, the notion that any of these pieces of cortex are in fact serving some isolated function is unrealistic given what we know about cooperative and competitive dynamics and nonlinear interactions in real brain systems. So the idea that you have a part, and if you take that part out, you have a window on the system doing everything else normally except for the part, is not consistent with most neurocomputational modeling. And language and reading, indeed, in living, breathing brains, involves complex cooperative and competitive dynamics. That's where information processing is. It's not in the pieces that can be selectively damaged. It's in the distributed, functionally connected operations, distinct parts of the brain working in sync and resonantly. That's where information processing occurs. And you can't get a lot of information on that. But new imaging technologies, such as those used by the center here and by all of our research centers, can be helpful. Two broad classes, I'm not going to spend time on this, but two broad classes of neuroimaging, functional imaging approaches: electrophysiological, which includes, I would simply include EEG and MEG as one class of studies that give you exquisite information on the timing of neural activity, but not necessarily such a clear view in their location. Most of you have heard this before. And hemodynamic, such as fMRI and PET, which actually give you very good localization, but because the BOLD signal, the blood oxygen level dependent signal, that is all the data I'm going to be showing you in this talk, is so slow to rise, about 6 seconds to peak, does not provide information about the timing of neural events, but provides exquisite information on location. It doesn't take a degree in rocket science to imagine that putting the two together is probably a good thing, because you want both time and space. 1, 2, 3, 4. So In this next slide, I want to show you a very basic study. It's not an old one, although it's not new anymore, that looked at typically developed adult readers. The study itself was actually a little more interesting than what I'm going to show you, because we had subjects either listening to sentences and making a judgment, are these good sentences or not, or reading the same sentences and making the same judgment, counterbalanced and blah, blah, blah. We also manipulated whether the sentence structures were object or subject relative in a way to get at syntactic complexity. And what we ended up doing was looking for those brain regions that are sensitive to syntactic processing demands, but don't care whether you read it or you heard it. That is, amodal syntactic regions. This was a study that Don Shankweiler, my colleague, was involved in, and he is very interested in sentence level processing. But for today's talk, here's what I want to stress. We had sentences come either to the ear or to the eye. And we're mapping out on this side of the screen. Oh, it's a little bit hard to see. I didn't realize it would be with the bright lights, but I guess you can see. That on this side, oh, and this is a very important thing which my translators and I are ready to tell you about. I'm showing this in radiological convention. So the left hemisphere is on the right side, and the right hemisphere is on the left side. So in this image, which starts, these are axial oblique slices. This is a lower slice. This is a higher slice. The left hemisphere is on this side, and the right hemisphere is on this side. It says if you're looking that way. Okay, so all the images I'll show you for the remainder of the talk, because except for a few that are 3D rendered, are going to be in this convention. So if it's on the right side of the brain, it's the left hemisphere. Okay? In this set of images here, what you're looking at are areas or voxels where there was reliably higher signal for speech, and if that's so, they're shown in purple. Now, it's not coming out so well, but those are purple and white. On the other hand, by t-test, we identified those voxels that are higher for print than speech, and those are shown in red to yellow. The first thing that you should note is that the areas that respond more to speech than print are located bilaterally along the perisylvian plane from the superior temporal and supramarginal gyrus to the inferior frontal gyrus. And there are some additional regions that are activated in parietal and frontal zones. But reading, the areas that respond more to the written form in these skilled readers are far more distributed through the brain. They include areas that are adjacent to the speech zones in both the temporal and the frontal areas. They include parietal regions and frontal regions, dorsal toward the top and ventral toward the bottom. It's a much more complex array of regions. And if you look on this set of analyses, this is looking at the same data in a slightly different way. It says, show me a voxel, which is this computer unit of analysis, that is significantly active for both kinds of modes, printed and spoken, irrespective of whether it was more or less in one or the other. That is, which areas in brain are multimodal? Because those areas that are active for both modes will also be good candidates for a system that begins to link speech and print together. And when we look at those regions, and we replicate a study that appeared a year or two earlier by Michael et al. From Carnegie Mellon, basically the overlap areas or the intersect areas are all in the left hemisphere. Remember, the left is on the right. And they include inferior frontal and temporoparietal and ventral occipitotemporal regions as well. And so this is a broad map of what the reading and listening circuits look like under some conditions in one silly experiment in one lab. But it's a pretty good overarching picture of reading versus speech. So when we consider what are the implications of this somewhat more distributed array of areas than the Wernicke-Geschwind model talks about, what are some of the implications? So the first thing is, and I'm not going to go into any detail on this, but those who've pursued the neural architecture for speech, for listening and speaking, have expanded upon the Wernicke's model in a number of ways. We still include the temporal and the inferior frontal areas, but there's been more stress on ventral, middle, and inferior temporal regions, or what Hickok and Peppel refer to as a ventral speech pathway, and some increased focus on a temporoparietal, premotor, motor, dorsal pathway. So unlike the simple diagram that we get from the Geschwind picture, a summary and interpretation of a lot of years of neuroimaging speech produces a somewhat more complex, but still largely left-dominant view of the listening circuit. There are some interesting action in the right hemisphere that they talk about at different temporal granularities that is beyond the scope of today's talk. But basically, we're getting a little bit more focus, and the simple intuition behind the Hickok and Peppel speech model is that when information comes to auditory cortex, it gets pushed in a number of directions. And when you need to be phonologically analytic, that is, to consider the nature of the speech stimulus, the phonological form, there's more involvement of the dorsal system. And the ventral areas for listening are more lexical, semantically constrained. It's a little more complicated than that for those of you who haven't read the papers. They're worth reading. Hickok's work is quite illuminating, especially the recent papers. But basically the idea here is that we have the basic architecture for spoken language. We have a dorsal and a ventral distinction having to do with more analytic versus more word recognition. What about reading, which is the focus of my research? Well, for a number of years now, we had sort of started out with a simple and embarrassingly underspecified initial model of the reading circuit based on what we knew around 2000, that in very general terms looks a little bit like the dorsal and ventral distinctions made by Hickok and Peppel, except that theirs was published 4 years later and didn't cite us. But other than that, I'm not complaining. But with regard to learning to read, we speculated that the temporoparietal areas, Wernicke's, angular gyrus, supramarginal gyrus, are a broader array of associative, heteromodal associative areas that are involved in linking orthography, phonology, morphology, and semantics, and that they're very much phonologically analytic. Guinevere had some studies as well showing that actual phonological awareness tests engage the dorsal to frontal areas, right? You had the sort of the temporoparietal and the premotor coming in to metaphonological processing. We also talked about an important role for the inferior frontal gyrus, which is involved in so much and has so many complex functions. But in regard to reading, we speculated that it plays one, particularly in a posterior aspect, is involved in mapping speech gestures into, mapping orthography into speech gestures. So it's sort of a motor-based component. And as you'll see in a minute, we also argued from this, our data, that the dorsal and anterior components of this left hemisphere reading circuit were critical in learning to crack the code. That is, in learning to make connections between print and language. Remember, you guys, again, I have nothing here to say on signing and reading. I'm trying to lay out what we have from speaking and reading as a foundation to see if it holds or doesn't hold at all as we do more research in signing. The third area that we emphasized, and certainly many labs did, and the work of Stan DeHaan and others has stood out, in Kathy Price as well, is the ventral pathway, which we argued is a late developing, linguistically structured, fast mapping system for print. In a nutshell, when you're learning to connect phonology, semantics to orthography, when you're trying to connect these things, the dorsal system plays a very important role in initial stages. And to the degree that learning is successful and you get more and more efficient, then there is the development over the first few years of literacy of a fast mapping word form system. It's been referred to in the literature as a visual word form area by some. Whatever the designation, there's good evidence that developmentally successful readers come to rely more and more on a fast mapping system into print. By the way, this is a bit of an aside, but there's also a lot of debate and question right now and good research, I know in your lab and others, on whether or not these kinds of taxonomies are going to be seen in languages such as Chinese and other languages that have very different ways of representing phonology in visual form. And I think that's an open and active area. But with regard to reading, these were the sort of broad categories. 2, 3, 4. So, yeah. Okay. So, this is a lot of information. What is it? So let me summarize simply. Typically, developing readers develop a good deal of specialization in the left hemisphere for reading. There's no doubt. There are distinctions between more dorsal and more ventral pathways developmentally. But there is also evidence that when reading is learned properly, you get a properly connected and functionally integrated, broadly three-part circuit. A frequent finding in studying children and young adults who with reading disability, that is, who never quite master or connect and crack the code, a frequent finding is underactivation of both the temporoparietal and also the occipitotemporal fast ventral word form area. This has been reported in many studies, in many languages. There may be variation depending on what tasks are used. But if you were to sort of do a meta-analysis, in fact, such meta-analyses have been done, you generally find evidence for left posterior anomaly, that is a lack of coherent activation, and as you'll see in a minute, connectivity in children and adults who present with reading disability. You often, but don't always, see a shift to the non-dominant hemisphere, and certainly our data showed it, and others' data shows depending. I think generally it's the case that if the task is demanding enough, you begin to see that shift. And, but you tend to see a shift to the right hemisphere and more heavy use of frontal lobe in general. Okay? So this is, if you will, the signature of reading disability, that these posterior regions are poorly organized for whatever reason, and fMRI doesn't tell you why in and of itself. We need something more. We need deeper tools. But there is a tendency to shift the circuit to the non-dominant hemisphere and to the frontal lobe. As I mentioned before, it's important not just to think about where some group is higher or lower in activation, or which piece of brain is active or not active when doing reading tasks, but actually to think about, from a systems level, how different distributed regions are either working in sync as a circuit or breaking down and not working as in sync. And so in a study that we published in 2000, this is old data, and we were preceded the year before by Barry Horowitz and Judy Rumsey published a very similar result we published in 2000. What we did is we looked at a large number of adult dyslexics or reading disabled folks versus controls. And we used something called functional connectivity analysis, which is asking whether two remote regions are operating in a correlated way, in sync. So to the degree that you have a system or a circuit, the activation in one region should be correlated with another. And to the degree that circuit is poorly constructed, there should be a breakdown in that correlation. And what we found, as did Horowitz and Rumsey, was that for the typically developing subjects, we found that the intercorrelations in the left hemisphere between the angular gyrus and these other regions, visual and language, were robust and highly correlated. But in the left hemisphere, they were in fact uncorrelated in the dyslexic adults. So it's not just that there was more or less activation, but these regions were not hanging together, working together. We found, as did Horowitz in a different analysis, that the right hemisphere connectivity between the right homologue of the angular gyrus, or the right angular gyrus, and the regions was actually stronger in dyslexics than control, again suggesting a shift to the non-dominant hemisphere. A brief word on longer time scale learning. We had an opportunity in studies we were doing at the Yale Child Studies, the Yale Center for the Study of Learning and Attention with Ben and Sally Shaywitz, to look at a very large number of children, 144 kids between the ages of 7 and 17, roughly half of whom were diagnosably dyslexic and half not. And the first question we were able to ask is, which brain regions would seem to play an increased role as you get older and more experienced, and which brain regions would seem to play a diminished role as you get older and more experienced? So this is a cross-sectional analysis to identify positive and negative correlations with age. And the most striking thing in this paper was that the left ventral visual word form area seen here was positively correlated with age in typically developing kids, which means the older you are, the more you have a relative shift to the ventral visual word form area. Whereas the right hemisphere and frontal lobe were negatively correlated. So that the older you, they're still involved in reading. You saw the reading circuit is distributed, but they're relatively less weighted, and the ventral more so. The dyslexic subjects, in the absence of successful treatment, showed a very different pattern. The older they were, the more they activated everywhere. There were no negative correlations. And the second thing we found was that since your ability to read and your age are positively correlated with one another, and both were positively correlated with this visual word form effect, which one is actually producing the effect? Is it maturation or skill? So using multiple regression, we were able to identify that age was not driving reading scores on a pseudo-word test were. And this shows the data from 144 kids around this ventral area. The higher the reading score, the higher the activation in a simple print task in the magnet. So the implication to us at the time was that for typically developing kids, as they learn the stuff of reading, the connections between orthography, morphology, and semantics, as all those things become apparent and get built into the system, relying on temporoparietal learning, there will be the training up of an expert system in the ventral visual pathway. There have been other studies that have shown this increased shift to the ventral pathway. James Booth, recent work by Jessica Church. There were some other data. I know that in Guinevere's study with, I always forget how to pronounce his name, he's such a great kid, Turkeltaub. Their analysis showed the reduction with age in the right hemisphere, but showed increases more in the middle temporal gyrus, I believe, with age. But I think that those data might not be consistent in that this ventral area and the middle temporal gyrus feed to one another, and there might be differences with regard to the measures we're using. The pattern of leftward shift and ventral, broadly ventral increase, I think holds across these studies. Certainly many others have come to show that. Now, why am I bothering you with this? Well, again, we're still just finally laying out what we expect to see if reading goes well, coherent, interconnected, three-part system, and how children who are struggling and not getting it tend to build an altered architecture. In another study, we asked the following question. This is an important, 1, 2, 3, 4. This is an important one for me, and it asks whether children, adolescents in this case, who are reading disabled and show this poorly organized left posterior cortex, whether these children have some access to these circuits, that is, that maybe these areas are noisy and unstable, but capable of being revealed. This is a crucial question if you're going to do intervention, because if you're going to do intervention, if the intervention is going to affect these poorly connected left posterior systems, right? If you're going to do treatment, you have to know whether there's latent function in there. Is there something there? And so, as one part of trying to understand the phenotype of reading disability, we did, and my translators are ready for this one, an incredibly dumb experiment, wonderfully dumb. And so what we did is to examine short time scale learning, we looked at the oldest trick in the book for making people get better at something, repeat stuff. So here was the basic design of this study. We have a group of typically developing adolescent readers and a group of severely RD readers. Okay? We were still using group designs. By the way, I don't use group designs anymore because I see reading disability as dimensional, and I see the power of regression over group analysis as a way to get at the brain mechanisms. But in any case, we're looking at group design. That was an aside, not for everyone in the room. And here's the simple study. While you're in the magnet, you're doing a very easy task. Is it living or non-living? Living or non-living? You're pressing a button, and unbeknownst to you, stimuli repeat up to 6 times in a run. So you're basically seeing the word 6 times, and once, twice, 3, 4, 5, 6. And pressing the button, yes, it's living if it's something like Tom, and no, it's non-living if it's something like the television. So it's a very simple and dumb experiment. It's also the oldest trick in the book for kids with reading disability to improve their reaction time and accuracy. If you repeat the word, they, like controls, get faster and more accurate. It would never constitute a good treatment for dyslexia because all you'd be learning was the things we repeated. So this is not a treatment study. This is a proto-treatment study, asking whether adolescent reading disabled kids have something waiting to come out that repetition might reveal. So the first thing we find, behaviorally, wow, something wrong with the, see, I knew that this kind of thing can happen, but I'm going to have to show you, the things got cut off. So this is reaction time with 6 repetitions, and in red are the dyslexics, which should say up here. And what you can see is, as you repeat the stimulus, you get faster. Whether you start out much faster, these are the controls in blue, or you start out very slow, you benefit from repetition. So the effects of repetition on reaction time and accuracy, this is number of errors, shows the same for both groups. It's an ordinal interaction, a little bit more for the dyslexics, but the same direction. However, when you look at the brain, The interaction between groups and repetition is entirely disordinal, which means it goes in the opposite direction. And so in all of these regions shown in yellow, which include all of the reading circuit areas, the supramarginal, the frontal, and importantly, the ventral visual word form area, we have a disordinal interaction, and it looks like this. Oh my God, how did this happen? You see, that's supposed to be the legend, and it should not have happened. Okay, so this is the word form area. This is the first two exposures, the second two, or third and fourth, fifth and sixth. What you see in the controls is what every study shows whenever you repeat stimuli in an fMRI experiment. As you keep repeating it, and it's getting easier, the bold signal, which starts out high, comes down. You're still using the area, but it works less, and so the physiological signal is coming down. No surprise, that's what happens. You repeat, it gets easier, the areas that process work less, there's less bold, less of an activation. What's happening in the dyslexics, this is the origin, it didn't come out here, is very interestingly different, right? So on the first four exposures, we replicate every published study, including our own, which is that they show low signal in the VWFA. In fact, they show deactivation. So this is what you find when you compare controls who have good activity to dyslexics. But after four repetitions, all of a sudden, ding, we have a response and a coherence in the VWFA. And the same is true in Wernicke's area and Broca's area. So what does this study imply, and why am I torturing you with it now? What it tells us is that A, repetition demonstrates that the circuit that we've been talking about as not developed is somehow hidden in that noise. And with a local boost of repetition, it reveals itself to be there. And so it suggests that if we could get to that system with appropriate intervention, we could boost it and keep it on. That's the hopeful message. It certainly suggests that the deficit, the problem is not of a sort of a developmental lesion, but rather a noisy system that even a local repetition will boost up. I hope you're understanding this. But it also has another implication which led me to a whole new line of research. We actually got an R01 this year to look at this issue, which is, it kind of stares at you and it says, wait a minute, why, for words that these kids have been reading since they were in first grade, did they need 3 local repetitions to reveal a circuit? Whereas the controls who start out, you know, they have a well-organized circuit, so it works hard at the beginning and it works less when it gets easier. By the way, all of this suggests a non-monotonic relation, a U-shaped relation between skill and activation, and really is a cautionary note for those doing neuroimaging of clinical populations. If you're just looking for where is one group high or low, my stupid study shows that if you happen to have repeated the items in practice, you'd fail to replicate 100 studies, because remember, by the end, the dyslexics have a higher signal. So it's not higher or lower. It's probably the heart of the phenotype is probably where you start and how you move through opportunities to learn. I'm arguing these days that the phenotype for learning disabilities is likely to be found in online learning and adaptation, circuit building and reorganization, not in where some region is either high or low. But that's one side. The more important point, this is telling us, it's smacking us in the face, because I know that if those kids come back tomorrow or 3 days from now, and I give them that same list, they won't activate until I give them a local boost. And the oldest clinical intuition in studying dyslexic children is that they're often bright, beautiful kids. They go home at the end of the day knowing everything that they need to know. They go to sleep, they come back the next day, and it's gone. It doesn't stick. And we'd call this consolidation failure. So we have a new R01 that I won't have time to, we have no data yet, but we're working to look at not just phonological or language or this or that, but actually how children with learning disabilities do or do not benefit from overnight and longer-term opportunities to consolidate. Consolidation failure is a very hot topic, as those of us who review a lot of grants can tell you, and it is so for a reason. But what we're really seeing is that there are problems in these kids, and Guinevere is smiling because she knew this way ahead of me, but there are problems in these children that need to be understood in terms of learning and memory and not just language. And I think that it's going to be a very important next step to look into this. So we're currently after this issue. Let me summarize all this so I can get to the last couple of topics. These data suggest a relation between skill and activation is complex and dynamic. Simple greater than or less than contrasts aren't going to cut it. And we need to really pay attention to learning and consolidation in any population that presents with difficulty. Now, I'm going to not spend any time on this, but there are a number of findings that have accumulated over the last 8 years or more showing that in those same left posterior regions, temporoparietal and less reliably but often the ventral occipitotemporal, there's also reduced gray matter volume and reduced white matter track coherence as indexed by diffusion tensor imaging. This is a broad and big literature. Eden's lab and others have contributed to this in both adults and children, showing that like the noisy, poorly organized, and improperly consolidated function, there's also reduced gray matter volume and reduced white matter connectivity in the left posterior region in children with dyslexia. And so to summarize all of that, so I can move on to the last couple of topics, functional and structural neuroimaging to date, in addressing the first few questions I wanted to raise and get out here so that we could ask how would this be different if your second language was English and your first language was sign? And if you did not have spoken language as the leverage you use to build the reading circuit, what would it be built from? I mean, this is, so again, I'm just trying to give you sort of an overview of current thinking on functional and structural neuroimaging. Functional and structural neuroimaging studies indicate that reading disabled children fail to organize left hemisphere regions into a coherent circuit. This includes unstable and reduced activation, reduced functional connectivity, problems in learning and consolidation of learning for new items, reduced gray matter volume, white matter track anomalies. This is just a wish list, not a wish list, a descriptive list, which gets me to the remainder of my talk. But all of this is describing what reading disability looks like in the brain. If I describe what it looks like in behavior, slow, labored, and error-prone word recognition is a defining feature. And it is often and almost universally accompanied by some problems in phonological processing, which may or may not be reducible to basic sensory motor mechanisms. Probably is to some degree. But these are describing and not explaining. How do we get to the next step in cognitive neuroscience such that neuroimaging is not simply following along from behavior, but has value added to behavioral studies? How can we get information that is not going to be gotten from behavioral studies alone? One important way is to move beyond mere identification of structural and functional biomarkers toward brain-based causal models of how these factors actually impede the development of this ventral specialization for print. Prospective longitudinal studies focused on gene-brain behavior pathways are at a premium, especially if those can start in preschool and watch literacy come online. Also at premium are studies that actually play with learning opportunities, what I was talking about with plasticity, where you could actually look at the brain in its different forms and different biomarkers, and then put children or adults through learning opportunities to see which patterns of anomaly in brain are most associated with which kinds of learning and consolidation difficulties in the context of language and literacy, or even in the context of non-language procedural and declarative learning and other mechanisms that many labs, including Guinevere's, have shown to be implicated in reading disability. By the way, this is muy importante. Kids who present with reading disability also show problems in things like statistical learning, in implicit learning, procedural learning tasks, the ability to notice if you're playing a simple reaction time task that things are repeating or not. And there are known brain pathways for procedural learning. They seem to be, in your data and other people's, compromised. And yet they're non-language. It has to do with, but may very well go to the ability to routinize and automate things and overlearn. There's less evidence, in fact none convincing, that there might be problems in declarative pathways in the brain, but certainly procedural pathways. I mention this, I have a mixed audience. Some of you follow this closely, some don't. But it's another area where if we're trying to understand why there are learning problems, we need to understand learning in a deeper way. So I'm going to present two things, because I'm almost to the end of my time. We are currently conducting 2 NICHD-funded longitudinal studies, and actually those who've seen me before, like Guinevere, I keep saying, you know, we're almost done, but we are now done this spring with the final cohort. So the next talk I give won't be on this sort of mining the data, but actually the outcome data on these children. We have about 60 kids left to collect. Oh shoot, that didn't come out either. Sorry guys. So there's a little thing here that says what kids do in this study. We recruit a large cohort of typically and atypically developing 7-year-olds. At time 1, we give them, we collect DNA, so we have genetics. We do structural and functional MRI. We do behavioral testing. And we also do something called magnetic resonance spectroscopy, or MRS. And then at time 2, 12 months later, the children come back and are given a behavioral testing, and at 12 months after that, behavior and brain. So it's tracking the influence of gene, neurochemistry, neuroanatomy, function on outcomes 24 months later in high-risk children. MRS, unfortunately, I won't have time to go into the data from MRS. I'll give you a very quick update. But this is a way of using an MR system to identify concentrations of important neurochemicals, like N-acetyl-aspartate and, very importantly, GABA and glutamate, the inhibitory and excitatory neurotransmitters, and choline and creatine, et cetera. And so this actually scans and reveals individual difference scores on each of these neurochemicals. We have them at time 1. Unfortunately, we've got them from a suboptimal part of the brain because that's all we could get. This is a large voxel in the visual cortex, but we got it. And now we're seeing whether those numbers, those individual different scores on expression early on, predict how well they read at time 3. So this is the design of the study, and I'm going to just mention 2 things. During fMRI, quickly, we have a task that is very simple and age-appropriate and allows us to map the initial circuit for print versus speech and words versus pseudo-words. This is a paper I sent last week. And so I'm not going to go into it in detail because I do want to just finish one last thing. But this was a study we did mining this longitudinal cohort, but we were interested in knowing whether children with putative speech motor difficulties very early on are developing reading and brain organization for reading differently. And so because we have a very large number of kids, over 150, that we're tracking in the study, We also have parental histories. And we identified, my young postdoc at the time, who's now in charge of the project at Haskins, John Preston, terrific kid, he identified 2 groups. He's a speech sound disorders expert. Kids who were late talkers and kids who were, well, 3 groups, late talkers, on time, and precocious. And we were very conservative because it's based on parent report. If the kid's not producing 2-word utterances by 3 and they went to see a speech therapist, these are kids who were, late talkers, even though retrospective parent memory data is not optimal. And there is a literature that is well established, actually, showing that children who are late talkers, again, this is all in the spoken area, are at elevated risk for reading difficulties later on. This has been shown by Luutinen in Finland, and Rescorler, and Scarborough, and others. So the first thing we did is we replicated this relationship in our data from the longitudinal study. These are children's time 1 visit data. And so what we find is on all of the reading and language assessments, late talkers are performing significantly worse than on-time or early talkers. But the critical finding in brain is this, that relative to on-time and early talkers, while either listening or reading simple words, the circuit that late talkers are using, and they're reading okay, sort of, and they're listening okay, but they had this early speech motor delay, which is still poorly understood in its etiology, but seems to be related to motor learning. These children are now reading and listening reasonably well, but with an incredible, almost a black hole of no activation for the putamen and the basal and caudate basal ganglia, the thalamus, and the superior temporal gyrus. So we literally have a circuit for both listening and reading that is poorly engaged the cortical-subcortical connections. And so this has a number of implications. One, these data suggest that late talking, along with its negative impact on reading, is associated with a failure to develop subcortical and cortical circuits that are critical in building an efficient system. And it implies an important but still poorly understood role of motor learning in the emergence of reading circuit, and may suggest an early biomarker for risk of learning and language problems. Okay, so I'm going to skip these. And I'm going to get to a summary of what we've done. I'm not going to show you each of the studies, but I'm happy to pass on the papers. Some are published, some are in press, some are under review. The only data that you're going to want to know about is going to be coming out next year, which is how did they end up 3 years later, 2 years later. And I hope to have some important information. But for now, we've examined brain activation at time 1 and time 2 and found late onset of talking is linked to subcortical anomalies in the basal ganglia and the thalamus. Problems with early reading readiness, and this is a study that I'm happy to pass on to you guys. It's in press in Brain and Language, or near in press. And it's a study where we took just the youngest kids from the longitudinal study, as young as we could get it. We got 63 kids who are basically 7-year-olds. And these kids are really beginning readers. And we asked a simple question. When we look at their reading circuit, kids who are doing better or worse on things like phonological awareness, temporal order judgments, rapid automatized naming, and all of these mix of skills, we can use those skills to isolate this beginning reading circuit. And what we find, in a nutshell, is that along with the expected areas in cortex, like low activation for poorly developing learners in the temporal parietal and the inferior frontal, we also find that poorly developing learners have anomalies in the pulvinar of the thalamus and in certain, and in the precuneus and other visual selective attention areas, and extending all the way back into visual cortex. So what we're finding from this, I wish I had had more time to go through it, so I'm just going to quickly summarize. We're finding that when you look at a reading who's actually just building the circuit, which is connecting vision and language, The cortical and subcortical sets of regions that are coming together include not only the language zones, but the visual areas, the fusiform gyrus, the pulvinar and the lateral aspects of thalamus, all needing to be coherently connected in order to crack the code. So I think that data is going to suggest just this. If you're looking at the emergent reader, you're going to find in any challenge, you're going to find anomalies in left cortical regions, but you have to pay attention to the subcortical regions. We also found that study, we have one finding, and again, I will just tell it to you and stop. The finding is this, that when we have fMRI predicting reading scores, and all of our labs do this all the time, you have scores of activation and scores of reading, and we get a nice correlation and a nice R-square when we simply predict how well you read from how well you activate the reading circuit. No problem. When we put in glutamate as one of the neurochemical indices, we get a 10% increase in R-square on reading. So all this is saying right now, and I have no story and nothing to tell you, but I want to make you aware, is this says there's value added in predicting reading variance from MR spectroscopy. We're trying to really track that down. We're looking at outcome data now to see how you predict long-term learning, but it suggests an important and understudied area of neurochemistry for looking at reading disabilities. And I'm going to end on this note. Many of us have done studies where we've looked at reading disability and response to treatment. The question is, with these weak and unstable and poorly interconnected and reduced gray matter volumetric and probably low glutamate systems, what are we going to do if we give kids a real opportunity to learn using the oldest treatment in the world that your grandmother invented? Practice, practice, practice. And so in a study that we published with Benita Blackman at Syracuse and the Shawitzes, I'm not going to go into detail. Many of you have seen this, and I will pass on the paper. But we looked at two groups of dyslexic children at a very young age, actually, just entering around 6.5 to 7. And there was a treatment group who got this. And this is what I mean by your grandmother's treatment. 50 minutes a day, 5 days a week, 9 months, reinforcing many of these basic component core dimensions. A dyslexic control group who were getting what the school gave them. We weren't withholding, but they were not. Actually, the treatment group came from Syracuse, where Benita Blackman was, and they flew 3 times to New Haven to do this simple fMRI test that I developed for this. And we found that the treatment group did much better. About 85% of the kids could be characterized as treatment responders, and that's real good, but about 15% not. And what we found in brain was that across the left hemisphere circuit, frontal, temporoparietal, and ventral occipitotemporal, we found robust increases in the treatment but not the control dyslexics. And we found that the front and the right showed after, because we scanned pre and then 12 months later post-imaging and then brought him back 1 year follow-up. And we got a monotonic decrease in the right hemisphere and a monotonic increase in the left with treatment. So it's a pretty good large-scale study. There have been a number of others that have been done more recently and in other labs. Temple et al. That used a fast-forward program showed left increases. But I want to summarize and end with this. There are a growing number of treatment studies that have shown modulation of these circuits with effective treatment. Functional changes, studies by Simos, Temple, Guinevere, Myler et al. And, very excitingly, changes in gray and white matter volumetric measures, studies by Eden's lab, I get Flowers et al., Keller et al. So all of these studies are converging on this. If you catch the thing right, you look at this noisy system, you impact it with good treatment, you can certainly see a lot of change. There's a lot of plasticity. There are a couple of things we should not be so proud of and we haven't dealt with yet. So one, what's optimal treatment for non-responders? In any good treatment study, even if you can get 80% of the kids to move, 15, 20% don't move. So we sort of have to begin to ask what works for whom. And we need to probably use experiments designs where we contrast multiple treatments against one another. And we don't ask which one is best on average, but which one works for which brain-based grouping. So there's a lot of work left to be done there. But we do have evidence that appropriate training has a normalizing effect on at-risk readers, that the left hemisphere system appears to be unstable but potentially trainable, at least in young children. We don't know much about what works for resistors. We don't know enough about kids with comorbid ADHD and other conditions, math, and we need designs that contrast multiple treatments. So in order to end, I want to end on the note that brings me here. First, I want to say thank you to my colleagues. And then I want to end on this note, and I hope that we have some time for questions. The story that I just put together, though oversimplified in trying to present it to a new audience, it sounds simpler and cooler and tighter than it really is. We have a long way to go. There's a lot of messy details about these systems. There's a better need to understand learning and consolidation. There's a better need to understand cortical and subcortical connections. There's so much we need to do. But I think we have a broad framework that actually is something of a success with regarding the brain for typical and atypical developing reading. We don't have enough explanation or causal models, but we have a pretty good description. And so I would end with the question, which is, if a child grows up in a signing world and has very natural, good language development using the same brain regions that support speech in hearing children, how do they make the double transition not only to literacy, but to English? And I think that that is a massive challenge, I'm sure you would all agree, to my simple story at every level. My story could fall apart, the hearing story, because it talks about using motor areas to ground and consolidate. It talks about using language to ground and consolidate phonological awareness development. How this occurs in deaf populations and signing populations is a massively important question. So I want to thank you for inviting me here. I hope that I've been able to give you a little bit of an overview of a complicated literature, and I look forward to working with all of you in the future. Thank you very much. Thank you. So do we have time for a few?
Maybe one or two or three.
Okay. Do we have? Yeah, Tom.
I'm gonna talk. Okay, I'll talk. Thank you. Training can change the brain. Is that?
Yeah.
That's what so, could the people who are late? To what degree do you think that people who are these late talkers and have these brain patterns that you've observed, is it a matter of exposure in the home? You know, say between the age of birth and 5, and so that if you can train it, I mean, is it a matter? Are you just providing the? Are you just providing the exposure that the child hasn't received in the home, and,
right.
And to what extent? Because there are many studies about the influence of socioeconomic status.
Yes, of course.
And social factors.
Yeah.
Kids not having vocabulary in the home, kids not having opportunity to gain these kinds of phonological knowledge
right.
in the home.
Right.
And so, is this a? Are we on? Okay.
Right.
Okay. Well.
So you're asking whether this is
are these biological purely, or are they environmental? Some kind of a combination between the two, and isn't it important to understand the home lives of these kids in terms of understanding what the possible brain trajectories are in terms of structure and function as it develops? Absolutely.
And I think that's a critical question for, certainly for our next priorities in research and what I wanted to talk to you guys about. We need to decouple environment from congenital. I think for a lot of the kids in my cohort, they came from very high SES families, highly stimulative environment, and there are significant numbers who will present with speech sound disorders ranging from late talking to stuttering to more severe SLI symptomology. We're trying to look at these kids. It is also the case that a lack of stimulation will inevitably lead to delay and to, and by the way, is an increased risk factor for reading disability, unsurprisingly. There are some researchers who are trying to pull them apart. Good work by Fumiko Heft and also by Kim Noble, who's at Columbia Presbyterian, and we're working with both of them, actually. But they're basically trying to pull it apart a little bit, because if you look at, and Guinevere, you might want to answer this as well, but there's certainly a suggestion that what presents in terms of atypical cortical organization is quite similar whether you got there from environment, you got there from prenatal exposure, or you got there from genes, at the level of reading and language. But there's got to be major differences in the underlying mechanisms, in the underlying involvement of subcortical areas, for example, in these different populations. Nobody, to my knowledge, has been able to pull them apart. And when they present in school, they present as poor reading and a rightward shift and the rest of it. So unpacking them is going to be critical to go to earlier stages, to look at the origins of literacy rather than trying to reconstruct what must have been there, as we did in this silly study. I'd rather be looking at 3-year-olds than looking at 7-year-olds and trying to guess backwards. So I think this is a super important question. It's relevant to all our research. That is, how does environment and congenital interact in complex ways? At this point, I would say that there is certainly nothing definitive I've seen from anybody. There's a little bit of this region goes more to SES and that region goes more to non-SES, but there's nothing definitive. But I think the story will be answered in birth to 5. That's where I think the story's answered. I don't know, Laura or Guinevere, others' thoughts on this one, but it's a super important area. Anybody? Okay. SES matters. Right.
Yes.
Is this one not working? Oh, it's working.
I was just jumping in to add that I would definitely, I see in my own,
People in the room might need to hear you.
just to say yes, that
Right,
the
right.
SES is extremely important, and we also see the very powerful impact of early exposure versus late exposure. Some of that we can see in first language exposure, but also in first exposure to a intensive bilingual context.
Right.
We see very fundamental changes in the brain and the engagement of that neurocircuitry, the recruitment of the neurocircuitry.
Right.
So,
Well, and of course, there's such important work in this, in the area of what used to be called critical periods and now seems to be morphing into sensitive periods. By the way, there's some incredibly interesting animal work that comes out of Takao Hensch's lab. I don't know if you've followed it, but it's worth looking at. He's working with Janet Worker and Chuck Nelson on some studies of that are looking at early visual and language development. But the key thing is that he's doing studies with mice where you can open or close these windows. It's extraordinary. Clearly, if you have a lack of stimulation beyond a certain point, you've got a downward trajectory, and what that's going to end up, what we're going to see in a 4 to 5-year-old, I think is still unclear. But Guinevere had her hand up. Go ahead. We have a lot of expertise in this room, so we should.
Well, first, let me say thank you for a wonderful presentation.
Thank you,
It's
Guinevere.
very inspiring, as always.
Thank you.
And maybe to add to that conversation to what Laura Anders mentioned, I think, you mentioned SES as being one variable. I think there's still many variables that we just don't know about. So there's work outside reading disability suggesting differences in brain structure in kids from low SES and high SES. Some of those brain structures are the same ones that you've shown are important
Yes.
contributing to the reading circuit, but the fields just haven't converged yet. I think some of us are guilty, you and us too, that we often study children from high SES.
Exactly.
And there's some investigators who are taking it on themselves to study kids with low SES, which is hard to do, but was very important.
Yeah.
So I think the effect of gender, I think, is still highly
Oh, yeah,
studied.
yeah.
This is a condition that's prevalent in boys, and so many of these studies are done primarily on male samples. I think there are a lot of questions there still to be answered.
I agree.
So I like this conversation and just wanted to add to it. But I wanted to see maybe if you could take a minute to tell us, you alluded to the fact that you were getting genetic data on your children, but you didn't have a chance to speak about that. Is there anything that you're willing to share with us at this point?
You know, I have slides that present, again, unsatisfying because it's just time one interconnections. So the paper that's under review, been reviewed and has now been resubmitted by Nicole Landy, and of course Elena Grigorenko, who's our geneticist on the project. The one thing, you know, we have a lot of data and we're waiting for the outcome data, which will be done by this summer. So basically the time 3 data. And then we have a number of statistical models that will look at candidate genes by MRS, by behavior, et cetera. So that stuff really will be done this year. But we've got the data, and she genotyped COMT, C-O-M-T, which is a gene that has been less clearly linked to reading, but has certainly been linked to attention, cognition, executive function, which is part of what's being trained up in reading. And what we have, the report we have, is the argument has been made that neurobiological data might be a more sensitive endophenotype for genetic grouping than complex behavior. And of course, the gorgeous work by what's his name? No, at NIH. Oh my God, I'm getting a senior moment. Who did all the studies with the genotyping and the fMRI of the amygdala? Oh my God. Anyway, this is some of the first studies to show that you get a better fit to bold of amygdala than to measures of anxiety on questionnaires. Oh wow, that's really scary. But in any case, so everyone is after candidate genes on behavior, on brain, on behavior. We'll have longitudinal data so we can do dynamic phenotyping. But here's what we got to date. We did COMT. We found that of course, the MET allele is the one that is associated with greater. Let me see if I can quickly pull that up. Just take a second. So we have the MET allele has been associated with higher dopamine levels in prefrontal cortex. So what we found is that when we group the MET, which is, this is a little esoteric question, but so this is a grouping based on, we combine the MET and the VAL-MET grouping allele against the VAL-VAL, which has been associated with lower dopaminergic expression in frontal lobe and worse behavior on executive function. So what we did is we found a reliable difference that the MET groups are doing better on all of our measures in this gene-based grouping. And so that just suggests a link to many of the behaviors we study. And the interesting thing, Guinevere, is the fMRI data, which shows, again, just shows a sort of an intuitive pattern. The same regions where good and poor readers differ, including in the STG and the OT and the IFG, a little bit more medial in the IFG, are showing this group difference. So here's the story. Where goes behavior, worse or better, there goes bold, and the only designator we used was this genetic grouping. So this is almost like an existence proof paper and not worthy of great, of great note yet. But it suggests that the bold behavior and the genetic grouping can hang together in a meaningful way in this age range, in this population. Of course, the real action comes now looking at candidate genes on behavioral outcomes. And there was one other thing we're trying to do with these data that's a little bit beyond today's discussion, but because we are able to quantify GABA and glutamate, we were particularly, Dr. Grigorenko is particularly interested in GABAergic genes, GABA family genes. And so we'll be able to look at the relation between polymorphisms there, GABA expression, and behavioral outcomes. But that work unfortunately awaits the final collection of data. I think it's exciting. I think that the other thing I'd say broadly, and we all know this, is that the power issues are ridiculously difficult. It has been difficult to show replicability from one to another. Ultimately, we know that we've got to get at the genetic level of analysis. I'm not sure that it is delivering an awful lot of R-square at present. And I think designs like this in broad sense that allow systematic connections across levels and over time so that the phenotype is dynamic, fingers crossed, might give us a better look into this. But in the meantime, we haven't even done any of the big candidates like DCDC2, and but those need to be done. So sorry, that was a long answer, but that's an exciting topic. Yes, of course. Thank you very much. Sorry to go on so long.
Sorry. Okay, thank you, Ken. That was great.
Thank you.
Everyone, thank you for coming and staying. It was a very stimulating conversation. So thank you.
Thank you, Tom.
for coming. Thank you for people who are still with us out there in the webland.
Oh, yeah.
What?
Hi.
So please do fill out your evaluation forms, fill them out, and give them to Connie over here. And thank you for coming, and we look forward to seeing you again 2 weeks, I think. Do we know? Soon. Connie, do you know when the next lecture is? February 15th. Yes, it's in 2 weeks from today. So we hope to see you then.
Can I put in a plug for the next lecturer? Is it Plout?
It is.
He is unbelievably good and smart, so you should hang out with him.
Yes. Okay. So thank you.
This has been a production of Gallaudet University.